Hello friends, in this video tutorial we will be studying about isolation of mutant. So what is mutant? Mutant is the organism in which mutation occur. What is mutation? Mutation is the change in the nucleotide sequence of a gene of a organism. Suppose here we have the gene having genetic sequence GG8ETC, and this gene is code for suppose protein A. If there is alternation of nucleotide like thymine to adenine, then the sequence will be GGA-TAC, and this will code for different type of protein which will change the total expression of the organism. So in case of microbiology, it has great effect that some bacteria have mutant to like some amino acid or some element, heavy metal, etc. So here we will be studying about isolation of mutant, and there are various mechanisms for isolation of mutant. One of them is replica plate method. So what is replica plate method? The mechanism behind the replica method is as follows. Okay, so first we have to take a petri dish. Okay, so we are taking a petri dish, then we are adding the media, and suppose we are wishing to separate a mutant which are auxotroph to leucine. So what is auxotroph? Auxotroph are the type of species which are specific to some kind of amino acid, protein, or some element, nutritional element. Without that they can't grow. Like for leucine auxotroph, the organism or bacteria can only grow in the presence of leucine. Without leucine it cannot grow. Okay, so for separating the leucine auxotroph, we have to follow this mechanism. Like we are taking a petri dish. In the petri dish we are taking the media like nutrient agar, and with that we are taking the leucine. Okay, after incubation of 24 hours, we will be getting the colonies like this 1 2 3 4 5. These are the colonies. Okay, so what we have to do, we have to replicate this plate. Okay, how? We have a disc shaped hammer. Okay, disc shaped hammer through which we have to copy this to two plate. Okay, how? We have to putting this and putting this and this. Okay, then what we have to modify these two petri plate, that we have to take media in both, but we have to add leucine in one, and we don't have to add leucine in another. That is, in one we have leucine and in another we don't have leucine. So what happened? The leucine auxotroph can only grow in presence of leucine. So without leucine they cannot grow. So what happened? So here in the presence of leucine we will find the exact copy of this. Okay, we have this like 1 2 3 4 5, and here it is also 1 2 3 4 5. But in case of absence of leucine, there may be variation if there is presence of leucine auxotroph bacteria. So those colonies which are auxotroph to leucine, they cannot grow because there is no leucine. So we don't expect growth for some colonies like 1 2 3 4 5. Here 5, in colony 5 there is no growth. So we can conclude from here that this is a leucine auxotroph. From this we can take and inoculate and isolate, and we can culture in a separate plate. By this, we can separate the auxotroph by replica plate method. And let me revise. So we have our petri plate. We are taking media and leucine, and after 24 hours, we will find the colonies, and from these colonies, we have to make two replica. Okay, both the replica plate, we have to take leucine in one, and we don't have to take leucine in another. In which leucine will present, it will be exact copy of the master media, and in another we will lack some colonies. Those colonies which are not present in media 2, okay, then we will compare it with 1, and we will conclude that those colonies are leucine isotopes. Like here, colony 5 is our leucine isotope. Thanks for watching. Keep subscribing and like and sharing, and we will study about another method of isolation of mutant in next video, that is resistance selection method.